So good afternoon, everybody. My name is Ed Conway. I'm the director of the Center for Blood Research. I'm pleased to be here. I have the easiest job. I just have to sort of hand over the baton to all these great summer students. Sort of the culmination of all their work for the last 3.5 months, and we're going to hear all about it, and then I'll be followed by a keynote speaker. But first, I want to thank particularly our sponsors, the Canadian Blood Services, the Sheldon Naiman and Linda Vickers Endowment Fund, CSL Behring, Bayer, Macropharma, the Neil Mackenzie Memorial Fund, Grifols, Novo Nordisk, Pfizer, Octapharma, Alexion, and Stego. These are all contributors to this program to support these students, so they get a little cash in hand and provides a unique opportunity. This is the first year the CBR has partnered with the School of Biomedical Engineering in this program. Peter Zanstra couldn't be here. I think at last I heard he was drinking wine in France. I can't understand why he's not here, but he sends his best, and I'm sure he'll be tuning in with a glass of wine in hand. Now, special thanks, of course, go to Hannah, and please make sure you thank them 1,000,000 times over, because really I'm just up here doing what they've told me to do in many ways. But Hannah Kim, who's the office manager for the CBR, Stephanie Mack, who's the director of the educational program, Kristen Fung, who's her assistant in the education program, and then a newcomer who's just arrived yesterday, so you'll see a lot of him over the coming year, is Andy Sue. They really did all the work. They put the program together. All the students know, certainly Stephanie and Kristen, for all the efforts that they've put out to make the program amazing, and I'm sure they can tell you more about it. So the day is really very straightforward. We have 2.5-minute presentations by all the students, so it's rolling right through, okay? So we have a giant stack of PowerPoint presentations here. They have 5 slides per person. They can't go over. If they go over in time, what happens, Kevin? Right, okay, so, and then you'll be turned off. After that, there's an open bar and posters and poster judging, and then a barbecue and awards. Now, the student presenters, I know you'll be a little anxious, I can see, but it's really cool in here, so that should help sort things out. Now, please, do not look back. I made this gigantamungo pointer here on the desktop here. As I said, the target is 2.5 minutes. You get pulled off at 3. Please look forward, not like that, okay? Because otherwise we won't see you, because it is being webcast. Now, you will be able to, all the students are going to vote for each other, okay? And you all have scorecards to vote for each other and vote for yourself. And then there'll be an opportunity. There's a People's Choice Award, and if you guys all have your phones, you can scan this Q mark down there, so you will be able to vote all the attendees, and that includes students as well. Otherwise, turn off your phones. Okay. In addition, you've seen these signs all over the place. If you want to post anything, everybody knows that pictures may be posted from this event. So with that, we're ready to start. The speakers, there's a handheld microphone, okay? And we can start going. It's on.
Okay. Never used a microphone.
You don't have to, and you don't have to, as long as you speak loudly up to the front, okay?
Oh, that's even worse.
Here's your, yeah, exactly, use that, okay?
Just
And
enter?
there's the- yep.
Okay.
Not enter, the forward.
Oh, alrighty. Okay, is time started? No, whatever, doesn't matter. Okay, so thanks for coming. Today I'll be talking about my lab's work in identifying the role of podocyclin in mouse mammary tumor invasion. So podocyclin is a transmembrane mucin that's closely related to CD34, and it's shown to be crucial for the formation of vasculature and the filtration slits of glomeruli. It's expressed in many carcinomas, and high expression has been correlated with very poor disease outcomes. Now, if you look at your right, I've got some tissue samples from colorectal carcinoma patients, and the sample that has high podocyclin, PODXL, you can see the presence of tumor buds, which is a cohesive cluster of fewer than 5 cells, and tumor buds have been shown to be closely related to collective invasion. And we see this present in the high podocyclin sample, and it's obviously not there in the low podo. So if we go to MCF7, which is a type of immortal cell line. We see the same sort of thing happen when we force high expression of podoclixin. There's these bud-like formations that have podoclixin localized to what we like to call a microlumen. And we repeated the experiment by using in vivo experiments with mice using a very common breast cancer model. And again, the same sort of thing. Podoclixin is stained in brown, and you can see it's present there in those microluminal formations. So the hypothesis here is that podoclixin expression promotes increased local collective tumor invasion in spontaneous mouse models of breast cancer. And that's pretty hard to say. But the aim of my work here was to develop a method for reliably identifying these tumor buds in mouse mammary glands. And that includes distinguishing between single cells and tumor buds, which can be either connected or isolated. If you look at the little diagram I've got here, you can see how a bud that might appear to be isolated in a two-dimension, like on a slide, can actually be connected when you visualize the 3D structure. So that's very important for us to do. The model, or the methods that we use, is very simple. We induce tumors in mice using the murine mammary tumor virus with the polyoma middle T antigen. It's a very common breast cancer model. And we made both wild-type and conditional podoclixin knockout mice with the knockout in mammary. So we harvested the tumors and fixed them, and we got to work analyzing them. We started with micro-CT, which is a kind of 3D visualization, very similar to what you might get if a doctor ordered a CT for you. And we got a lot of success with this, but the main problem is that the tumor bud has very similar radiopacity to normal mammary duct tissue. So without some kind of other marker, it's virtually impossible to tell what's tumor and what's not. We need some sort of context. So we started sectioning. And sectioning is very good because we can stain for polyoma middle T, which is present in this model in all cancerous cells. And we saw a lot of success with that, but now we still need to go back and look at some 3D models to see if they're connected or isolated. And that's where the work is today, where we're using some software to do that reconstruction from these sections. And that's all she wrote.
Okay, hello everyone, my name is Amardeep and my project is called the PASSES 3D study. Regulatory requirements in North America mandate that pre-transfusion compatibility testing must be done on patient blood samples that are less than 3 to 4 days old. Pre-transfusion testing involves a group screen which determines the patient's blood group and screens the patient's blood for the presence of alloantibodies which are formed as a result of sensitizing events. Sensitizing events are the exposure to foreign red blood cell antigens as a result of previous transfusions or pregnancies. At the Vancouver Coastal Health Authority, inpatient order forms for blood products allow physicians to repeatedly order grouping screens every 3 days to ensure in-date testing for the provision of cross-matched blood. This is to reduce the occurrence of adverse transfusion reactions, which are often associated with mortality and morbidity. With this in mind, the primary purpose of the study is to question why the 3-day rule for grouping screens is an option at VGH, despite patients not having any sensitizing events during the course of their hospital stay, or being at low risk for transfusion where serial grouping screens are not necessary. This will be done through the primary objective, which is to determine the rate of positive antibody detection tests, or ADTs, for alloimmunization on a patient cohort hospitalized at VGH. Out of a total of 980 patients receiving grouping screens every 3 days in 2018, blood transfusion data, which is obtained from the laboratory information system, was analyzed for 350 patients in the interim. Out of these 350 individuals, 6 patients tested positive for alloantibody during the course of their hospital stay, with 3 patients testing positive for a clinically significant alloantibody. All of these patients had a previously documented history of transfusion, except one which tested positive for a clinically insignificant alloantibody. 9 patients tested positive for an alloantibody for their first AT of their hospital stay, except for 1 which tested positive for an autoantibody. 16% of the 350 patients did not receive transfusions despite receiving serial grouping screens every 3 days. If the final data is consistent with the interim analysis, this shows that there is a possibility of changing the 3-day rule for grouping screens, as less than 2% of the patients tested positive for an alloantibody during their hospital stay, typically with a documented transfusion history. Waiving the need for serial grouping screens every 3 days reduces costs and excessive blood collection, which is part of a patient blood management strategy that has been found to improve patient outcomes. Thank you.
Good afternoon, everyone. My name is Aya Zakaria. I work for Dr. Helen Cote, and today I will be discussing the increase of mitochondrial and cellular toxicities first-line HIV regimens. So HIV infects CD4-expressing PBMCs, which are usually CD4-expressing T cells. Therefore, people living with HIV receive a lifelong treatment with combination antiretroviral therapy, also known as CART. CART suppresses the viral load by inhibiting specific stages of the viral life cycle. Each CART regimen contains 2 nucleoside reverse transcriptase inhibitors forming the NRTI backbone, and 1 base drug of the following classes, all color-coded for your convenience. As important as they are to HIV treatment, certain CART regimens are associated with mitochondrial and cellular toxicity, but the effects on human PBMCs are poorly understood. And the long-term safety of integrase inhibitors, which are first-line regimens now used today, are relatively unknown. In CD4 T cells, ex vivo exposure of dolutegravir has been shown to impair mitochondrial function and dampen overall immune response. Therefore, we aim to characterize CART-related cytotoxicity and mitochondrial toxicity in PBMCs, and to compare the effects of different integrase inhibitors, specifically raltegravir and dolutegravir. So we isolated PBMCs from 6 healthy donors and cultured them in CART-containing media at 1C max, which is maximum pharmacological concentration in plasma. And after 6 days in culture, we performed flow cytometry using the following probes to quantify apoptosis, viability, and mitochondrial markers of toxicity. These graphs show the data from all six subjects, each shown in a unique color and normalized to their own DMSO drug solvent control. You can see in the purple boxes, which highlight normalized total cellular apoptosis and normalized mitochondrial reactive oxygen species, which is a mitochondrial marker of toxicity, that dolutegravir-containing regimens elevate levels of apoptosis and mitochondrial ROS, whereas raltegravir-containing regimens do not. To summarize, in terms of most of the CART regimens that I used above, lopinavir-containing regimens and dolutegravir-containing regimens increase apoptosis or cellular toxicity and mitochondrial toxicity, whereas raltegravir does not. So comparing different integrase inhibitors, dolutegravir seems to have a more toxic effect. So please stop by poster number 5 for just more discussion about first-line HIV regimens or just a fun chat. Thank you.
I'm Brenda, and this summer I was working with my supervisor Tanya at Dr. Karen Chung's lab, where we were working on optimizing GELMA-based hydrogel for cell growth and morphology in a microfluidic chip. So, the chip we were working on was an airway on a chip, and it's made by 2 PMS layers with the perfusible lumen running throughout the chip. So this chip is meant to be a physiologically relevant model of the human airway. Within the channel, we have a monolayer of human airway epithelial cells on top of a biodegradable PLA membrane. Below that, we have our GELMA hydrogel, where fibroblasts are within that, and then we have the perfusible lumen with human umbilical vein endothelial cells binding it. This experiment focused on optimizing this GELMA hydrogel for the fibroblasts within it, and we wanted to see proper elongation as that's part of good morphology. Our first experiment was testing the GELMA and LAP cross-linker percentages, which are the 2 ingredients that go into making the hydrogel. The LAP is a photo-initiated cross-linker, so when exposed to UV, it'll solidify the GELMA. As you can see, in the 8% GELMA, we have a lot of red, which indicates cell death. So if you look at the 6% GELMA, in the yellow box, that is the concentration that we picked, as it showed proper morphology growth and also created a stable enough gel for a perfusible lumen. The next experiment we did was exploring additives in the hydrogel. So we included collagen and Matrigel that was sonicated for 45 minutes, as these are shown to be great for fibroblast growth. And as you can see in the experiment with the GELMA and Matrigel, we saw elongation within 2 days, and after another 3 days, we saw continued growth and elongation. We have another experiment with media that we didn't have time to show, but feel free to come to poster 7 to check that out. But from this experiment, we were able to determine that a 6% GELMA and 0.6% LAP mixture with 30 seconds of UV exposure was ideal for our experiment. The addition of Matrigel led to better physiologically accurate cell morphology. The next steps would be to optimize the hydrogel for 1HAEs and Hubex to create the functional triculture model. And finally, when we have the functional model ready and that can replicate physiological responses, we'd like to use that to study COPD, which is and drug response. Thank you.
Hello. Okay. Hi everyone, I'm Cameron from the Kim Lab. And my project this summer looked at integrin receptors in platelets and their interactions with a cytoskeletal protein called filament A. So integrins are found on many different kinds of cells, but they're especially important when it comes to hemostasis, or the way that our body stops to bleed. So as a quick breakdown, primary hemostasis involves three main steps in platelet adhesion, activation, and aggregation. And integrins play a role in each of these steps. For example, beta-1 integrins are important for adhering platelets to collagen, and even more importantly, the beta-3 integrin alpha-2b beta-3 is essential to platelet aggregation, as it can cross-link multiple platelets together to form a plug, which will eventually stop the bleeding at the injury site. So my project focused on this integrin alpha-2b beta-3 and its signaling process in platelets. So before a platelet can aggregate, these integrin receptors must first become activated. And to do that, we need a very specific set of signaling reactions called inside-out signals, and these can be triggered by agonists like collagen. So these signals will eventually lead to the binding of regulating proteins to the integrin receptor, which causes an activating shape change, allowing it to bind its ligand. So filament A is believed to be one of these regulating proteins that interacts with integrins in a way that prevents their activation, and our lab is curious about how this proposed role fits both into platelet signaling and function. So using Crelox recombination, we generated mice without filament A in their platelets. Then we treated their platelets with a range of agonist concentrations. And then lastly, we used flow cytometry to compare the knockout and control responses. So first we looked at P-selectin binding, which is shown on the graph on your left. And from that, we can see that the platelets are not activating properly without filament A. Next, we were curious about integrin alpha-2b beta-3 activation. So we used an antibody specific for the active structure of this receptor. And from the graph on your right, we can see that similarly, filament A is required for normal beta-3 integrin activation. We're also curious about how platelet function was affected without filament. So we repeated a similar experiment where we took platelets from the control and knockout mice, except this time we let them sit in a glass tube after simulation, just to see how well they form clots after time. So from the image on the left, we can see that up to an hour after simulation, the platelet response, the difference in platelet response is quite striking, with the knockouts barely even aggregating. And then we quantified this difference in aggregation using light absorption measurements, and that's shown from the plot on your right. So altogether, we've shown that filament A is required for platelet activation and platelet aggregation. And the next steps we have to take are to try and figure out which signaling molecules filament A interacts with to better establish its role and function in platelets and hemostasis. Thank you for listening.
Hello. Hi, my name is Kristen, and I'm the current Education Program Assistant at the Center for Blood Research, and I work closely with Hannah and Stephanie as part of the admin team. So today I'll be presenting something very different from the other summer students. So get ready to learn about what I did for CBR social media. So I do manage all the social media platforms, but today, just to make things simpler, I'll only be discussing Twitter. And I'd like to, through analyzing some tweets, I hope to share with you guys why Twitter is a great platform for scientists like yourself. So first off, I'd like to mention that I use Hootsuite to publish all my posts. Hootsuite is great because you can manage your content, manage all the social media channels, and also schedule your posts. So for the first post that I'll be talking about, this is about new research from the Castro Lab about self-propelled particles. And so this is on your left. And then to further promote this tweet, I collaborated with the Michael Smith Labs social media coordinator about their blog posts. So for the third tweet that I posted, as you can see on your left, I linked the original publication just in case audience members would like to dive even deeper into the research and also get some more background context. And for the last tweet that I posted in this series, as you can see on your right, this is a member profile about the publication's lead author. And I did this to showcase the human side of research and also to showcase that there is life outside of research. So just to showcase my results and the success of these tweets, let's move on to Twitter analytics. So once I add up all the impressions of these tweets, impressions meaning the number of times that people have viewed this tweet, the number is over 5,000 total views, which I think is pretty impressive. So the moral of this story is that if you have any research that you you'd like to share with me, you should send it over so I can feature it on our social media platforms. So social media is obviously not the only thing that I work on. I'm also responsible for event planning, and I'm excited to announce that I'll be planning a CBR dog therapy event for late August. So if you want more updates on that, and also more updates on the Center for Blood Research, you should follow us on the accounts I listed to your right. Thank you.
Hey everyone, I'm Jillian from the Jeffries Lab, and I'm going to be talking to you today about knocking out EHD4 and EHD1 in a dendritic cell line using CRISPR. CRISPR is a type of adaptive immunity found in microbes that is now being used as a gene editing tool in mammalian cells. It uses a single guide RNA to target a specific sequence of a genome, and then recruits Cas9, a nuclease, to introduce a double-stranded break in the genome. The cell is obviously going to try and repair this break, however this is not done perfectly and can result in permanent mutations in the genome. The purpose of this project is to determine if EHD1 and or EHD4 play a role in dendritic cell cross-presentation. Our lab has found that EHD1 and EHD4 interact with the MHC class I molecule H2KB, which is involved in dendritic cell cross-presentation. And my specific role was to knock out EHD4 and EHD1 in a dendritic cell line using CRISPR. So first is test if our single guide RNA and our CRISPR technology was actually creating a mutation in the EHD4 or EHD1 gene. We performed a surveyor nucleus assay, which uses a nuclease that cuts when there's a DNA mismatch, such as when the CRISPR technology has inserted a mutation in the gene. This will result in cleavage of the PCR products, resulting in multiple bands, as highlighted by the red boxes. Next, to test if our mutations were actually causing knockout of the EHD4 or EHD1 genes. We took the colonies that passed the surveyor nucleus assay, and we subject them to Western blotting for EHD4 or EHD1, and we found that we were able to get clones that had knocked out expression of EHD4 and EHD1. So overall, we were able to create dendritic cell lines with EHD4 and EHD1 knocked out using CRISPR. This will allow us to perform different functional studies, such as cross-presentation assays on these dendritic cells, and we'd also like to use similar techniques to fluorescently tag EHD4 and EHD1 in dendritic cell lines to see what proteins they are interacting with and if they are actually interacting with the H2KB molecule. Thank you.
So good afternoon. My name is Hani Ibrahimi, and I will be presenting my summer work on the effect of protein kinase inhibitors on function and storage quality of human platelets. Recent findings regarding the impact of protein synthesis within platelets have shown in the two publications you can see, has opened new opportunities for further understanding for platelet functions. Initially, selected platelet protein inhibitors are used to further understand the effect of their protein kinase targets on storage and to later pick an inhibitor as a viable candidate for future experiments. As for my method, first, pooled buffalo platelet concentrates from NetCat were allocated into small bags spiked with inhibitors. Next, in vitro quality assessments were done on storage days 3 and 8. After we performed Western blots targeting proteins related to platelet storage quality. Lastly, after we performed Western blots, the target proteins related to platelet storage quality, After we performed Western blots that targeted proteins that are related to platelet storage quality. And lastly, mass spec was performed to monitor protein synthesis of the treatments. Results for mass spec and Western blots are not completed yet, and they're still being processed. my results, there are two in vitro quality parameters, as you can see, lactate levels and ADP response. Many more results can be found on my poster, on poster number 11. But in general, inhibitors have exhibited different impacts on storage quality. And lastly, once all the protein analysis results are obtained, only then, with the correlation of in vitro quality results, can we provide a novel conclusion that leads to an inhibitor to be picked as a viable candidate for future experiments. Thank you for your time.
Hello. Hi, my name is Emma, and I've been working at Simple Lab to study the mechanism of concussion over the summer, and I'll be presenting on finite element modeling of brain strains during soccer entries. So soccer is the only sport that uses unprotected head as the primary contact of the ball, and there are controversies over the possibility of TBI due to soccer heading. And finite element modeling are used to investigate the mechanism of TBI, and the two epi models that I've used for the study are called Simon and PCBRA. The major difference between these two models is that Simon takes kinematics data and computes the strain results, having high computational costs, whereas PCBRA has its own built-in lookup table to interpolate for the strain results. So the objective of my project was to investigate the potential for TBI using Simon and PCBRA during soccer errors. Prior to the experiment, we made participant-specific mouth guard with IMU sensor attached that measures kinematics data, and each participant performed 30 headers in total, 5 at the front, left, and side at 2 different speeds. And after the experiment, we ran the data on Simon and PCBRA to compare the strings and their locations. For the results, for linear acceleration of around 2G, Simon and PCBRA both had peak strain less than 5%, meaning that the axonal damage can be recovered. However, as shown in the figures, the peak strain locations were indicated at different places. In conclusion, for our experimental data of linear acceleration around 2G, the peak strains were found to be less than 5%, meaning the axons are in transient depolarization stage and they can be recovered. In the future, we'll run more experiments with higher rotational accelerations and linear accelerations, and we will also utilize another epi model to validate the results. For Simon and PCBRA to be used in this type of study, we will look into reducing the running time of Simon and figuring out the method to identify the exact location of interest. Thank you.
Hi everyone, my name is Henry West and I was with the Crystal Lab this summer. My project was on the initiation of coagulation on HIV. Thanks to recent advances in combination drug therapy, patients infected with HIV can now live long lives. However, they still have a 12 times greater risk of thrombosis. This and related pathologies are associated with many enveloped viruses. The viral envelope is comprised of host-derived lipids and proteins, as well as viral encoded proteins. It has been shown on other viruses that the envelope can acquire tissue factor, which acts as a cofactor for the factor 7A mediated cleavage of factor 10 to factor 10A, leading to coagulation and cell modulation. Through these mechanisms, tissue factor on the envelope of viruses contributes to enhanced infection and associated pathologies. This project aimed to study the biological activity of tissue factor in the envelope of purified HIV and assess its effects on plasma coagulation. The virus was purified by ultracentrifugation through a sucrose cushion, greatly increasing the urine concentration as shown by the P24 Western blot analysis. Electron microscopy revealed particles in the size, range, and shape as expected for HIV.
Assays were performed to assess the ability of the virus to activate factor 10 by cleaving a chromogenic substrate to factor 10A. A titratable effect was observed, with the greatest effect shown at the highest concentration of HIV. NAPC2 and the antibody, both known to block the function of tissue factor, both significantly reduce factor 10A generation. Results of the coagulation assay were in line with those shown by the chromogenic assay. For more detail, please come see me at my poster. Overall, this project has shown that purified HIV initiates coagulation in a tissue factor-like, factor 7A dependent manner. Positive identification of the virus particles as HIV, as well as confirmation that tissue factor is actually associated with them, is still required, and investigations are currently ongoing. However, this preliminary data still provides a possible explanation for the thrombotic pathologies associated with HIV, as well as provides further support for the development of antiviral drugs targeting tissue factor. Thank you.
Hi everyone, my name is Jennifer and I've been working with the MAW lab this summer on determining changes in red blood cell deformability that occur during blood storage. So first of all, you're probably wondering, what is red blood cell deformability and why is it important? Red blood cell deformability describes the ability of a red blood cell to bend and squeeze through parts of the microvasculature, which might be up to 4 times smaller in diameter than a red blood cell itself. We already know that red blood cells become less deformable during storage, and this loss of red blood cell deformability has been considered a biomarker for the quality of blood bag units. One of the fundamental issues in transfusion medicine is that not all blood bags are able to deliver the same benefit to transfusion patients, and that's what we're trying to fix. So we're using a microfluidic Batchett device to sort red blood cells based on their deformability, and we predict that if we can sort red blood cells based on their deformability, we can identify donors that might be able to provide a longer circulating red blood cell, which provide a great benefit to chronic transfusion patients. So the microfluidic Batchett device works by pushing red blood cells upwards through the sorting region through constrictions of decreasing size. The red blood cells are going to be sorted based on their ability to deform through these constrictions, and the sorted red blood cells will end up in one of 12 distinct outlets. The results of this study have shown us that there is a donor-to-donor variability in the initial red blood cell deformability, as well as the rate of degradation of this deformability throughout storage. So I performed experiments on donor 9, as you can see up here, who shows red blood cells that maintain their integrity very well throughout storage in comparison to some other donors that were analyzed earlier in the study, such as donor 1. So all in all, we can conclude from this study that different donors will provide red blood cell samples of a different initial deformability, as well as different rate of degradation in their red blood cells. This provides a lot of room for future studies, such as a marine transfusion model and maybe monitoring the red blood cell deformability in a blood bag. Thanks for listening, and come visit me at poster 16.
Good afternoon, I'm Jennifer Laird, and this summer I worked with the Orbit Group on the synthesis and characterization of a potential bifunctional chelator. In 2018, approximately 1 in 6 deaths was due to cancer, according to the World Health Organization. The medicinal nuclear chemists approached this global health crisis through the development of promising new radiopharmaceuticals with the goal of both early detection and therapy of cancer. Radiometals offer desirable therapeutic and/or diagnostic properties depending on the decay characteristics they exhibit, while a suitable bioconjugate such as an antibody allows for the selective delivery of this radiometal to disease cells. The molecule responsible for linking these two essential functions is called a bifunctional chelator, and it must possess unique characteristics that will allow it to form a stable metal complex that will remain intact when delivered into the blood. This summer, I focused on optimizing synthesis of the potential bifunctional chelator H2 amino HOX. Notably, the gold standard ligand donut is a macrocyclic chelator that requires high temperatures and long radio labeling times in order to form a stable metal complex. However, more mild conditions are expected for H2 amino HOX due to its non-macrocyclic and rigid structure. These mild conditions are very desirable because they offer a promising route towards radiopharmaceuticals that are both convenient for clinical applications as well as are more compatible with temperature-sensitive targeting vectors. So through varying the base, temperature, and solvent, I was able to decrease byproduct formation and increase the formation of this product by 8%. And efforts are currently underway in order to investigate whether metal complexes can form under mild conditions with non-radioactive metals in order to investigate which radioactive metal complexes are worth pursuing. So successful synthesis of H2 amino HOX lays the groundwork for extensive future studies with this chelator, particularly in gaining insight into the metal complex structure as well as its potential behavior in the blood. If you're interested to learn more, please stop by poster number 17 and have a conversation. Thank you.
Hello everyone, my name is Jessica Ha. Growing up, I watched my grandma injecting insulin into her body before every single meal. She has type 2 diabetes, a disease that can lead to major organ failure. It's also associated with insulin resistance and obesity. So Dr. Conway's lab have found that mice lacking CD248 are protected against type 2 diabetes and obesity. CD248 is a glycoprotein expressed on fat tissues, but we are wondering what's the mechanism of CD248. So we need an in vitro model to study it. My aim for the summer was to develop a 3D spheroid model using cells from fat tissues of mice with and without CD248. So I collected the fat tissues from mice, extracted the cells, and plated them in the non-coherent 96-wall plates. The cells, they aggregate after 4 days and form spheroids. Then I add in the differentiation media to induce the formation of mature adipocytes. Here we have the image of the spheroid throughout the 4 weeks. At the top, we have the CD248 wild type, ND, non-differentiated, and D, differentiated. And at the bottom, we have the CD248 knockout, non-differentiated, and differentiated. Throughout the 4 weeks, we see that the non-differentiated spheroids, they stay relatively the same size, whereas the differentiated spheroids, they increasingly grow in size due to the development of mature adipocytes that accumulates lipid droplets. Then I took the area measurements of those spheroids. Here we have at week 3, the CD248 knockout differentiated spheroids are much larger than the CD248 wild type differentiated spheroids. This corresponds to our in vivo model. Thus, we have established an in vitro model to study the mechanism of CD248 in type 2 diabetes. Thank you very much. If you have any questions, feel free to come see me at poster 18.
Hi everyone, my name is John, and I'm part of the Digital Health Innovation Lab. So my research is on perception of professionals and their users on trust of artificial intelligence for medical decision making and risk predictions. So right now, AI is very popular in the field of medicine. It seems to hold great promises in reducing healthcare expenses as well as improving overall health outcomes of patients. And it's often developed using something like machine learning, where as you can see in the figure, there's a lot of medical information that can be fed into a system for it to develop So the method is fairly straightforward. We hope to distribute surveys to both the physician population as well as the general public to see how they view AI being used in medicine. So here's a small snippet of the survey that we've developed. So it's scenario based, and there's two scenarios, one low risk and one high risk. So the red box indicates the effect or the generated metrics by the AI technology, and the red arrow indicates what the AI recommends in individualized cases. So basically, we hope to see what key stakeholders feel regarding the choices that they're given, whether they prefer the general statistics or the generated AI statistics that is generated. So currently the survey is still being distributed, so we don't have any results, but we hope that once the results are in, the pilot study can help inform our recommendation on how to increase acceptability of AI technology. Thank you.
Hi everyone, I'm Jonathan with the Wallace Lab within the Department of Electrical and Computer Engineering here at UC. And today I'm going to talk to you about automating image analysis for the structural assessment of 3D printed cardiac tissues. So just a little background, the Wallace Laboratory recently pioneered this technology called lab on a printer, through which self-containing vitamins flow through a microfluidic mixing device, and then also directly integrated inkjet dispenser that allows us to kind of pattern these fibers into 3D structures. And currently we're working with Aspect Biosystems to commercialize this into a platform for high throughput drug screening studies on the printed cardiac tissue. So my goal is to develop a custom image analysis program that can automatically quantify the structure and maturation of these printed tissues. There are a couple unique complications that come along with cardiac tissue, specifically because cardiomyocytes are often binucleated, and upon imaging they seem to be really highly irregular. In terms of methods, start off with just a literature review of established segmentation methods, and then over the course of a few months, alongside a lot of extensive testing and iteration, we developed an algorithm and implemented it in Cell Profiler. So our resulting program was able to successfully segment all the images in our test data set, along with produce these measurements here that are of interest to us. And looking ahead, I'm currently writing a standalone application in Python that will also implement additional analysis for specific protein stains like dystrophin or maybe connexin 48. And also we're going to be implementing support for subcellular analysis, which is essentially sarcomere and set bandwidths, which we're very interested in. So I'm going to be at poster 21. I haven't bored you to death, so feel free to stop by. Thank you.
Hey everyone, my name is Kieran. I'm from the Hancock Lab, and my job this summer was studying the role of universal stress response proteins in the survival of Pseudomonas aeruginosa. So Pseudomonas aeruginosa is such a bad bug when it comes to infecting cystic fibrosis patients and immunocompromised that the World Health Organization has deemed it of critical priority to develop new antimicrobial treatments. The human body can be a stressful environment to infect, whether it be the immune response of the human body or the various pHs that you'll find. Bugs that colonize the human body will have their work cut out for them. Lucky for Pseudomonas aeruginosa, it does encode for 7 universal stress proteins. We know that universal stress proteins are produced under suboptimal growth conditions, and we know that they increase the stress tolerance of bacteria. In the context of Pseudomonas, however, what I want to find out is whether there's a survival advantage in a wild type over a heptamutant with all 7 of these USP genes knocked out. And then from there, I can complement back individual universal stress protein genes into the heptamutant and see if individual genes are associated with a given form of stress. So one form of stress that I was interested in is pH. Bacteria will come across a variety of different pHs within the human body, certainly in the gastrointestinal tract. So essentially what I wanted to do was stress my cells with pH 11 growth medium for an hour. So that's comparing the wild type to the heptamutant and all of my complement strains. And so sure enough, the heptamutant was survival deficient compared to the wild type. They're at 100%, the heptamutant you'll see in blue. And some of the complement strains there in orange, you can see that they did restore that survival phenotype, particularly 56, 590, and 66, 460 over there on the right. And another form of stress that I'm interested in is when invading human body bacteria may get engulfed by macrophages. So essentially I'm able to monitor the cytotoxicity of Pseudomonas toward macrophages by looking at lactate dehydrogenase release. So what I found was that wild-type Pseudomonas aeruginosa is the most cytotoxic of the bunch compared to the mutants, which is, again, fit my expectations. And I did see that the heptamutant was less cytotoxic. And with the orange, again, as you see in the orange complementations, essentially we restored some of that cytotoxicity. So in summary, Pseudomonas can have its survival under stress boosted by the presence of USP genes. I did look at some other forms of stress, which I did present here, but you can have a look at them on my poster number 22. Thank you.
Hi everyone, I'm Matt Josh, and I'm part of the Rossi Lab, and this summer I worked with Maureen to investigate the role of TAC1 in muscle-resident FAPs. Well, what are FAPs? FAPs are cells that are contained within muscle that help in successful regeneration of muscle. These FAPs proliferate when muscle damage occurs. In states of chronic damage, like seen in dystrophic patients, these FAPs begin differentiating into adipocytes, or fat cells, and fibroblasts, which are harmful for regenerating muscle. The fate of these FAPs is determined or depends on the immune cells that surround it. These immune cells can activate and inactivate different cellular pathways that are involved in the FAPs. One such cellular transducer is transforming TGF-beta activated kinase 1, or TAC1. And in order to investigate the role of TAC1 within FAPs, we can delete it using the HIC1-Cre recombinant system. Here, something called tamoxifen can be injected into the mice, causing the knockout of a target gene, in our case, the TAC1 gene. After this, muscle damage can be induced, and then we can evaluate the regeneration through methods like the cross-sectional area of muscle fibers or the number of nuclei per fiber. Prior to my project, it was found that TAC1 knockout mice actually had a decreased muscle fiber size compared to the control. In addition to this, there was actually an increased recruitment of immune cells called eosinophils to the muscle. However, these experiments were done in the acute damage state, so only 7 to 14 days after damage had occurred. My project focused on chronic damage, that is, using 6-month-old dystrophic mice to better model the human disease. However, we did not find any significant change in the muscle fiber size, and we didn't find any significant change in the number of eosinophils recruited to the muscle. This suggests that perhaps TAC1 only has a role in the acute setting, or perhaps that the system had been exhausted by the time we took these measurements at 6 months. When we took a step back and looked back at the TAC1 knockout FAPs in the acute setting, we found that there was a high expression of a cytokine called CCL11. And perhaps this cytokine is actually involved in the recruitment of eosinophils. In the future, a migration assay can be used to test this hypothesis, and perhaps it will give us insight into why eosinophils recruited was so much in the acute setting, but not as much in the chronic setting. Thank you for listening.
I guess I don't have to go today.
Well, that's maybe the next
I don't think so.
maybe I hope so. I don't know. They just cut you out.
That's okay, I don't have to talk.
But you have to sing instead.
No.
No, no? Okay, wait a second. Let me just make sure you're here.
Oh, there.
go take a rest. We'll call you back up. Okay? She'll be back.
No.
Yes, yes. So first I want to just make sure is Dr. Butler in the crowd here. Is Dr. Butler here? Hi, Dr. Butler. Just checking. Okay. We're just going to take a little stretching break here for 1 minute. First, let's thank those first, 14, 15 people. So before we continue for the next group of students' presentations, we're just going to talk a little bit about Neil Mackenzie. Neil Mackenzie, on January 11th, 2015, Neil Mackenzie, a postdoc in Dieter Brahma's lab, who's a PI here in the Center for Blood Research, died tragically in a climbing accident at Joffrey Peak, just in north of northern British Columbia. Neil was really a remarkably unique person. He passionately embraced life in and out of the lab. He grew up in Scotland. He did his PhD there and a postdoc in Scotland, and then really wanted to come to Canada, to Western Canada, to become a climbing instructor. And so he came, but he was sort of balanced between loving the great outdoors and loving science, and he was really intrigued with the work that Dieter was doing in his lab. So he joined the lab, and he was really on his way. He was making remarkable advances in investigations in cardiovascular disease. But all his extra time was spent outside, canoeing and everything you can do in British Columbia, he was doing it, but particularly climbing. And he was a member of the Varsity Outdoor Club, where he was instrumental in sort of bringing young people out, teaching them, mentoring them, which is what he was doing in the lab as well. He was sort of the embodiment of what a mentor should be. And I give you a list of what was, and mentoring comes in different flavors and sizes and shapes, and it's really how you define it, but these are sort of descriptions of what a mentor is. It really came from those who voted for this awardee. And so when Neil died, one of the things that came out was that this was something that he really loved to do, was to support his peers and younger people. So we established this annual award, and this is an award that's voted by students, trainees, anybody, to recognize the support given by anybody, your peers, and those who support you outside of PIs. And we always have a whole slew of applicants. The awardees, or the nominees are judged by old friends of Neil's, so that's outside of our hands largely. These are the names of the nominees this year. We always have several, and this isn't to say that we don't have lots more great mentors within the Center for Blood Research, but these are the ones that were nominated, and you should all be honored by that place. But this year the awardee is Dr. George Butler. And George says, come on down, George. So we had to fly George in. She was on vacation. I'm not sure what she was doing, but she wasn't in the lab, to be sure. But I think I've just introduced you just recently at another event. Is that right? Anyway, so George received her Bachelor of Science at the University of Kent in Canterbury in the UK, did biochemistry studies, and then spent a year in Italy, and then did a PhD at the University of Leicester. And then moved to the Strangeway Lab, research lab in Cambridge, a very famous lab, to do a postdoc. and then to the University of East Anglia, which is where the lab moved at that time. In January of 1999, she came here for 2 years. And she stayed. And we're really glad that she did, because she's been really a stalwart supporter of her mates in the overall lab, and there have been many over the years, because it's a big lab. For other CBR labs, so she's really seen the move from other sites to this building, and she's been a major supporter of her peers and the more junior people, and there are several more junior people here who can attest to that. She really gives her all, putting them first, promoting their careers, facilitating all the time. She's a lovely person and a great mentor, and one who's very deserving of this award. So, George.
Thank you. Well, I did write a few things down.
Okay, here.
My son told me that if I read this, I would be able to enter CringeCon immediately. So he told me not to say that, but I'm still going to read them. Okay, so 5 reasons why I mentor students. Firstly, I like to watch their confidence grow as they're trusted to go for it. Secondly, I like to watch them fail. Not because I'm a sadist, but because it's always good to watch people realize that science doesn't always work first time. When you do experiments in a lab as an undergraduate, the experiments are designed to work. But really, it's that I enjoy watching them get up, try again. I really enjoy it when somebody discovers something that nobody else in the whole world knows. Watching them go from, oh my God, I've got no results, to having a story to tell for your poster or your talk or your report. The 5th reason is really selfish, I guess. Not because having a student is a cheap source of labour, because to be honest, it's a huge effort to supervise and mentor somebody properly, and oftentimes it's actually quicker to do it yourself. But that's not the point. It's actually a really good feeling to kindle an interest in science for somebody. And this is not one of the 5 points, but last but not least, you can't actually mentor somebody by yourself. So I'd like to acknowledge the overall lab over the years, because when I agree to mentor somebody, it usually involves roping in numerous other members of the lab who know more about a particular topic than I do. So it's really a group effort to mentor a student. That's it.
Where's our photographer? Oh, there you are. Okay, so I'm going to just take that off because it'll. Okay, so a plaque. It's a plaque, and it's the Center for Blood Research Neil Mackenzie Mentorship Excellence Award presented to Dr. Georgina S. Butler. And this plaque is a very special plaque because it's made here in Vancouver by a spin-off from the University of British Columbia, and these are chopsticks. Made from chopsticks. Do you know how many chopsticks are thrown out every day in Vancouver? Any guesses? Come on, some guesses.
3 million.
3 million. Okay, so let's go a little lower. 100,000 are thrown out every day. So this company collects the chopsticks and recycles them. They collect about 60% of them throughout Vancouver, and they make amazing things. Okay, so we work with them. Okay, come on over here.
My chopping board.
It's a plaque, and you can use it. Yeah,
It goes with my chopping board.
Okay, you can go on vacation. Yeah, really. Okay, we're going to call on Meredith Clerk again. Okay. You're up.
Almost got away with it.
Yeah, nice job.
All right, hello everybody. So my name is Meredith, and I've been working in the Kids lab for the past few months. And today I'm going to talk to you a little bit about endothelial cell growth under shear stress and how we can use this to model physiological vasculature. Okay, so first of all, what is shear stress and why do we even care about this? So a lot of cells, endothelial cells in particular, are undergoing flow every day in our bodies.
And we can actually see a lot of differences in cells that are grown under flow versus those that are grown under static conditions. And the main difference that I'm going to talk to you about today is in the glycocalyx structure. So the glycocalyx is really important, and it plays a big role in a lot of things like cell-to-cell communication and even like activation of immune response. The thing is, if we want to study this under physiologically relevant conditions, that means studying it under flow conditions, and so that brings us to microfluidics. So right now in our lab, we are currently experimenting with two different microfluidic methods. The first is our homemade chip, and the second is the bioflux. So we've been able to produce a lot of biomedical experiments on our homemade chip, and this has been fantastic, but the problem is it is really time consuming. It takes about a week to make one of these things, and you can only use it one time. So in search for a higher throughput method, we've recently been experimenting with the bioflux, which is really, really neat because it allows us to do these same flow experiments, but in the convenient, well-played format that we're all used to in cell culture. So if you look at the figure on your left here, you'll see glycocalyx grown under varying shear rates. And basically what this is showing you is that with higher shear stress, we're actually seeing an increase in the glycocalyx structure. And then on your right, you'll see an endothelial monolayer that we're actually able to grow using the bioflux instrument. Now, like I said, this is an ongoing project, but we were able to get the cells to adhere, and we were able to see glycocalyx structure after only 1 day in the microfluidic channel. So in the future, we are going to be replicating all of the biomedical experiments that we have currently done on our microfluidic chip in the bioflux. And this is really, really exciting moving forward because it's basically another step forward in making sure that all of our future cell experiments are that much more physiologically relevant. So yeah, if you want to hear more, I definitely want to tell you more. Please come see me at Poster 28. Thank you so much.
Hi everyone, my name is Nathan. I had the pleasure of working in the overall lab this summer, and so the title of my project was Cholesterol Regulation by Membrane Type 1 Matrix Metalloproteinase in MDA-MB231 breast cancer cells. So just a bit of an introduction. MT1-MMP is a zinc-dependent transmembrane MMP involved in degradation of the extracellular matrix, and as well plays an essential role in cell migration. It also promotes tumor invasion and metastasis. So with this, I was looking at its relationship to PCSK9, which is known to reduce LDL cholesterol uptake by promoting LDL receptor degradation, and is actually a candidate substrate for MT1-MMP. Knowing this all, we hypothesized that the proteolytic processing of PCSK9 by MT1-MMP contributes to MDA-MB231 breast cancer cell migration through cholesterol regulation. So one of our major methods of analysis was an LDL cholesterol uptake assay. So what we did was incubate cells with LDL cholesterol labeled with the pH red fluorophore, harvest these cells, and then perform flow cytometry analysis on the BD FACS Canto. Before performing this actual assay, though, we wanted to confirm protein overexpression of MT1-MMP and PCSK9. So we did this by Western blot, and so you can see in the doubly expressing line that the PCSK9 levels were lower compared to some of the other lines, and this implies cleavage by MT1-MMP. On the actual LDL cholesterol uptake assay, you can see that there are significantly lower levels of LDL cholesterol uptake compared to its vector control. And when inhibiting MT1-MMP with myrmestat, we saw significantly higher levels of LDL cholesterol uptake. Next, we wanted to assess cell migration in response to MT1-MMP and PCSK9 overexpression, so we did this by performing a scratch wound healing assay. And we saw that over a 48-hour period, all 6 of our stable cell lines had closed the lesion that was made at the time 0 point. Interestingly, it appears that the PCSK9 expressing lines actually had much easier detachment compared to their vector controls. So to conclude, what we saw was that MT1-MMP overexpression reduced LDL cholesterol uptake. We saw that when inhibiting MT1-MMP, there were significantly higher levels of LDL cholesterol uptake, and we validated that our scratch assay actually worked. For future direction, we want to perform our scratch assay again, as well as performing a cell membrane cholesterol assay after an LDL cholesterol treatment, plus or minus an MMP inhibitor. Thank you very much, and come find me at Poster 29.
Hi, my name is Olivia, and I'm a summer student in the Kazakh Adaptive Lab working on the cryopreservation of red blood cells using novel polymers. Every year in the US, there's 30 million units of blood required for transfusion patients, but meeting this need life cycle of cells and inadequate storage methods for these cells. Cryopreservation is a common approach used to store cells, but there are limitations for the cryoprotecting agents, such as ice recrystallization, which puts stress on the structure of the cells, and cytotoxicity. So we hypothesize that our highly hydrated branched polymers will protect the cell's integrity better in cold storage. So we first aim to quantify the ice recrystallization ability of our polymer and to test the biocompatibility of our polymer with red blood cells using hemolysis studies. So first, for the ice recrystallization inhibition, we quantified this by using the ice crystal radius as a function of time, and we analyzed this using LSW theory, and we were able to derive the ice recrystallization constant from this. The value for HPO is lower than that of other polymers, which aligns with the prediction that it is able to lower the diffusion between ice crystals. You can see in this image that the HPO has more of a distinct circular shape, while the control they're merging together, so that's the diffusion between the ice crystals, which puts stress on the cells. For our hemolysis studies, the value for HPO, which is in the blue box, you can see is very comparable to current industry standards, glycerol, and then it's even lower than AGS, which is also a standard. This is an important result because it's showing that in the future, with the aim of therapeutic use, that it is biocompatible. So overall, we were able to quantify the ice recrystallization inhibition ability of our polymers and to show that it is biocompatible with the red blood cells. In the future, we want to improve our wash methods after we take them out of the liquid nitrogen based on their osmotic fragility. And we also want to expand our concept to nucleated cells, including stem cells and T cells. Thank you.
My name is Paige, and I work at the Photomedicine Institute with Daniel Louie as my supervisor. Imagine you're at the beach and you see a mole on your shin. It's new. You go to the GP, get it checked out, and they say that it looks suspicious, so you should go see a specialist. Oh, great. It's an 18-week wait until the next opening. 18 weeks later, you go to the specialist, they take a biopsy, and the results are going to be another 2 weeks. 2 weeks later, you get the results, and you are positive for melanoma. This is the deadliest skin cancer type. And your survival rate may have dropped from 97% to 15% in that time. 20 weeks sounds like an exaggeration, but right now the average wait time for referral to treatment is 23.2 weeks. And as you can see from the red plot, it is steadily increasing since 2009. My lab is developing a low-cost, handheld design to detect skin cancer. It's for the hands of your GP, and it's going to detect skin cancer within milliseconds. It uses a laser beam, which is a uniform polarized light. When it hits your skin, it scatters, and then when it depolarizes, and the way that it depolarizes is how we detect skin cancer. There's a few things before we can get to commercialization, and that is what I'm doing. So in addition to making it functional, we want to make sure that it increases manufacturability, so that's reducing the number of components, making it easier to use in a clinical setting, and so we're going to make it more ergonomic and more portable, and we're going to up reliability by adding a centering and aiming design. And here is the design right here. In the center, you can see the aiming and centering features. To the right, you can see the ergonomic package with all the components in one neat package. And there is an exploded view on the left side. And to meet the specifications, I have decreased the number of components by 4. I've added a twist-on attachments, and I've decreased the size by 5. We are currently testing the we tested geometry and now it's going to the clinic, so this is the future of skin cancer detection. Thank you. Come see me at those degree too.
Hi, my name is Parveen, and this summer I worked in the Divine Lab to study the impact of infusion pumps on the quality of transfused red blood cells. There are many advantages to using infusion pumps for transfusing blood products, such as the ability to precisely control the flow rate. However, the extramechanical stress on the red blood cells and the resulting hemolysis is a clinical concern. Currently, there is a wide variety of pumps used across Canada, and there is no standardized testing protocol for quality assurance methods. So, we are investigating exactly what impact these pumps are having on our transfused blood products. We hypothesize that different pumping mechanisms and flow rates will have an effect on the quality of the infused red cells, and the magnitude of this effect will vary depending on the age of the blood. So in order to conduct this study, we received 3 ABO matched units per repeat, and these were pooled and split into 4 transfer bags, each of which were transfused using a different pump model currently used in Canada. So we collected samples from the baseline, which was the pooled bag originally, and then from gravity infusion and flow rates of 50, 150, and 300 milliliters per hour. The collected samples were evaluated based on 5 different parameters. The first of which is hemolysis, and we discovered that there is a pump-related increase of hemolysis. So the change in hemolysis was calculated by subtracting the baseline level of hemolysis from the percent hemolysis caused in each of the pump samples. And here we can see that in day 22, for the 22-day-old blood samples, we have an increased hemolysis for the peristaltic pump type compared to the piston type at the rate of 50 milliliters per hour. And for 40-day-old blood, we have a significantly higher increase in hemolysis for each of the rates for the peristaltic pump type compared to its gravity, as well as an increased hemolysis for the peristaltic pump type compared to the linear peristaltic pump for 50 milliliters per hour. Moreover, the increase in percent hemolysis is significantly higher in 40-day-old RCCs compared to 22-day-old RCCs. Another parameter that is of clinical relevance is potassium levels, and we noticed no significant impact of the pumps on the potassium levels compared to baseline. And if you would like to learn about the other parameters which we measured and the results we gained, please visit me at poster number 33. Thank you.
Hi guys, I'm Pavneet, and today I'll be talking about developing a chronic infection model with Pseudomonas and human bronchial epithelial cells. So Pseudomonas is one of the dominant bacteria to colonize in the lower respiratory tract of immunocompromised patients, which is actually sterile in healthy individuals. For example, 80% of cystic fibrosis patients will succumb to respiratory failure due to chronic bacterial infection. So once again, what I'm trying to do is remodel this bacterial infection in the lower respiratory tract, but I'm simplifying it quite a bit because I'm working with just one cell line, so the human bronchial epithelial cells or HBE cells. So as you can see, they're kind of smooth and elongated in shape, but when you infect them with wild-type Pseudomonas, within 6 to 8 hours, this will lead to killing the host HBE cells. But this 6 to 8 hour infection time isn't really representative of a clinical chronic or severe infection, and that's what we want to move towards. So one way to increase infection time is working with mutants, and that's what I'm doing. So I'm working with 3 different mutants that have gene deletions in virulence factors, which are the secretion systems. So what I did was I infected 100 bacteria per HBE cell, And I was able to quantify for cellular cytotoxicity or cellular death plotted on the y-axis after conducting something called an LDH assay. So as expected, the wild type in black has about 70% cellular cytotoxicity, and so does one of the mutants in blue. More interestingly, 2 of the mutants represented in orange have a 3-fold reduction in cellular cytotoxicity. And in addition, in the images below, you can see that the cells are more intact compared to the wild type. So basically, we were able to show that we can increase infection time from 6 hours to 18 hours, and the cells are more viable. The greater impact of this is once you build an in vitro chronic infection model, this can serve as a preliminary model to test novel therapeutics, and then you can move up to more complex and expensive models. Some other things we were interested in were how these mutants compare to the wild type and what immune response is induced when you infect these mutants to the host HB cells. So please come check out my poster if you have time. Thank you for listening.
Hi, my name is Rogan, and this summer I had the honor of working with Dr. Haley Merkley and Dr. Antonis at the hemoglobinopathy program of British Columbia, where we looked at documented immunization rates in asplenic patients of the adult hemoglobinopathy program. So asplenia is defined as the absence of the spleen anatomically or functionally, and this is really common in sickle cell disease patients and thalassemia patients, putting them at overwhelming risk for overwhelming post-pulmonary immune infection. Infections such as Streptococcus pneumoniae with encapsulated organisms such as Streptococcus pneumoniae, Neisseria meningitidis, and Haemophilus influenza. And since these patients are also frequent users of blood products, they are also at risk for developing infection with hepatitis B. And so international guidelines recommend that these patients get immunized and also receive prophylactic antibiotics. So this is a translational study, which means that we wanted to assess the gap between the current standard of care and actual real-life practice. So we wanted to determine the proportion of asplenic patients in the HPDC with documented immunizations meeting international guideline standards. So we did that first by identifying asplenic patients in the HPDC. Then we did a retrospective chart review, and we carried out surveys to family physicians for the patients that we did not have any records for, and then we analyzed our data. So there are currently 143 actively enrolled patients in the HPDC, 94 of which are asplenic, and a whopping majority were, it turns out, not vaccinated up to international guidelines standards. So we also wanted to look out, so there are a total of 5 vaccinations. We wanted to see who received maybe 3 out of 5 or 2 out of 5 of the vaccinations, and how many had no documented immunizations at all. And it turns out that there are around 53%, so even if they received vaccinations, how good are vaccines if not documented? In conclusion, there is clearly a lack of documentation of immunization records of asplenic patients in the HPDC. Unfortunately, we weren't able to collaborate with the Vancouver CDC or the public health records, and it's really difficult to obtain complete vaccination records from family physicians. We made tons of calls. There may be a role, hopefully, the study will be able to lobby for doing immunizations in the HPDC for asplenic patients. And this study really proves what we already know, that implementing the knowledge that we already have is as important as coming up with new science. Thank you for listening.
Good afternoon, everyone. My name is Samantha Marr, and I'm a college student at the Carson Lab. Today, I'll be talking about a project that investigates IGF-1R as a potential therapeutic target for treating lenalidomide-resistant del(5q) MDS. So patients with myelodysplastic syndromes, MDS, have abnormal blood cell development and are associated with poor outcomes. Around 15% of MDS patients have interstitial deletions on chromosome 5q, and also known as del(5q). And around 60% of del(5q) patients are responding to first-line therapy lenalidomide. It is believed that the haploinsufficiency of casein kinase 1A1, which is a gene on chromosome 5q, confers sensitivity to lenalidomide in del(5q) MDS patients. However, 50% of responders eventually become resistant. So there is great interest in finding alternative targets of del(5q) MDS. Closely located next to casein kinase 1A1 are microRNAs 143 and 145, and their haploinsufficiencies have been shown to play an important role in MDS. IGF-1R is a direct target of these microRNAs in solid tumors, thus the loss of these microRNAs de-repress the expression of IGF-1R in these models. IGF-1R pathway has also been predicted to be a target of these microRNAs in MDS based on del(5q) expression profiles. So this leads us to our main question: does microRNA 143, 145 haploinsufficiency confer sensitivity to IGF-1R inhibition in del(5q) models? In order to test this, I use colony-forming assay to test in vitro sensitivity to IGF-1R chemical inhibitor in a myeloid cell line haploinsufficient for microRNA 143 and 145 KG1A. From our results, cells treated with the IGF-1R inhibitor show significantly reduced clonogenic output compared to control, suggesting KG1A is sensitive to the IGF-1R inhibitor. So to conclude, we found evidence to support microRNA 143, 145 haploinsufficient cell line, KG1A is sensitive to IGF-1R inhibition. In the future, we will repeat the same assay in transfused human cord blood and in lenalidomide-resistant cell lines. Thank you.
Hello, I'm Shannon from the McNaghy Lab, and I'll be presenting today on podocyclin's role in ovarian carcinoma. So a little bit of background: podocyclin is a cell surface stem cell protein related to CD34, and it's involved in cell adhesion and cell-to-cell junctions. Podocyclin has been heavily implicated in poor patient prognosis in several different cancers, and our lab has previously shown that high podocyclin expression results in increased tumor growth and increased metastasis. In most cancers, podocyclin is present in a small percentage of the tumor cells, but in ovarian cancer, podocyclin is present in a large percentage of the cells. But as of yet, there's been no publications relating the functional role of podocyclin in ovarian carcinoma. So our hypothesis was that knocking out podocyclin in an ovarian cancer leads to a more epithelial-like monolayer and decreased tumor growth. To do this, our first aim was to compare cell-to-cell junctions between podocyclin knockout and wild-type cancer carcinoma cells, and second, to compare tumor growth in vivo ovarian cancer xenograft model using the same 3 cells. So first, using an immunofluorescence, we used F-actin to stain the actin cytoskeleton in green, and red in ZO1 to stain the tight junction protein 1. And as you can see in the wild-type cells, they are very disorganized, and whereas compared to the podocyclin knockouts, they're much more organized, epithelial-like monolayer with no holes. And next, we looked at wild-type tumors in vivo, and you can see on the left that the wild-type tumors grow much quicker than the knockouts. And then on the right, you can see that tumor weight after resection is significantly higher in the wild-type when compared to the podocyclin knockout tube. So to conclude, the results suggest that the presence of podocyclin in ovarian carcinoma changes cell-to-cell junctions and promotes tumor growth. So to continue to investigate the functional role of podocyclin in ovarian carcinoma, we're currently investigating in vivo structural podocyclin mutants, and we'll also be looking at an anti-podocyclin antibody in ovarian cancer because it was previously shown to reduce tumor growth and reduce metastasis in breast cancer. So we'll see if it has a similar effects in ovarian cancer. That's everything. Thank you for listening, and my poster is number 38.
fluorescent stem cell and leukemia. So as you can see from my topic, I'm working on a gene called MYC, which is a polar oncogene that is found to be frequently overexpressed in many forms of cancer and hematological malignancies, including acute myeloid leukemia. So my project is built upon using the existing model, MYC model of leukemiogenesis that were made in our lab, which is we forced overexpression of MYC in normal human hematopoietic cells and turning them into leukemic cells. Then we would inject the cells into immunodeficient mouse, which produces human growth factor, and to monitor to see if human myeloid leukemia develops. So we use this model to study disease initiation and progression of AML. So in the previous experiments, we use the model and we inject with many cells. However, AML is known to be a clonal disease, which is arise from a single cell. So we were wondering, can we use a single MYC overexpressing cell to generate leukemia in the mouse model? We transduced primitive cells with MYC virus and sorted them and injected them into irradiated immunodeficient mouse. And as a result, as the frequency calculation suggested that 1 in 14 of the cells has leukemia initiating ability. Therefore, as a result, 1 in 16 of the transplanted mice we got leukemia. And on the flow analysis on the left, we can see there are large numbers of human and GFP positive cells found in the mouse bone marrow. And the majority of them are positive for CD33, which is a myeloid marker, indicating that the leukemia we produced was indeed human myeloid leukemia. So for future steps, we could use this model to further investigate the biological and molecular mechanism of the disease, since it's shown that it has the ability to give rise to leukemia from single cells, which better recapitulate the real human AML. Thank you.
Hello everyone, I am Sia and I did my research project in the McNagley lab in the Biomedical Research Center. A simple action like breathing requires muscle motion and can cause muscle damage. So our muscles are constantly being repaired and regenerated, and our immune systems plays a crucial role in this process. Now imagine every time you breathe, instead of muscle repair, your muscles waste away and are replaced by fat and fibrotic material. Well, this is what happens in kids with the genetic disease Duchenne muscular dystrophy. They're wheelchair-bound in their teens and dead before their lives can begin. But what if we could manipulate the immune system, which is out of whack due to the disease, as a possible therapy? The inflammatory profile for muscles shows that immune cells play a crucial role in the regeneration process, especially eosinophils, which have also been shown to play a key role in the allergic response initiated by type 2 or Th2 immune cells. The overlap in these two immune processes allowed us to investigate whether being Th2 skewed or allergic promoted better muscle health. To test this hypothesis, we used vancomycin as an antibiotic treatment to induce Th2 skewing in a mice model for muscular dystrophy. Th2 skewing is characterized by high levels of IgE and eosinophils. Lifelong vancomycin treatment induced high levels of eosinophils in the muscles, shown here in red, and high levels of IgE in the serum. To further assess disease severity, we quantified fiber size, with smaller fiber size associated with worse disease prognosis. And even though we did not find a significant difference between our control and our vancomycin treatment group, in our vancomycin treatment group, we found that IgE levels negatively correlated with fiber size, with higher IgE levels equaling less and worse disease outcome, and lower IgE levels equaling better disease outcome. In summary, I have shown you that vancomycin-induced Th2 skewing created IgE-dependent fiber size, and this has several implications for the microbiome because it has been previously shown that antibiotic treatment using vancomycin changes the nature of the microbiome, and this could affect muscle regeneration. If you'd like to hear more about this and other future Th2 skewing strategies that we plan to use in our study, please visit poster number 40. Thank you.
Is anyone else disappointed we didn't get to hear the kazoo? Because I was looking forward to that. My name is Arthur Gavanchi, and I'm a summer student in the Zenstra lab. So hematopoietic stem cells, or HSCs, are present in umbilical cord blood and are an attractive candidate for cellular therapy. This is because they're able to reconstitute the entire blood and immune system. And compared to bone marrow, they have less stringent HLA matching. However, cord blood does have some downsides, the main one being the lack of neutrophil and platelet engraftment. Below is shown one of the current outlines for a cord blood transplants. What we're really interested in is between day 1 and day 7, where we're able to manipulate the cellular population to adjust to improve patient outcomes. My first goal was to develop an automation pipeline for manipulating the HSC population. To do this, I had to ensure that my experimental design was as solid as possible. That's why I went with, oh, I'm so tired. That's why I went with a factorial design. And factorial designs allow us to test out multiple factors at once, and in addition, also allow us to test out interactions. In addition, the current outline is using CD34 positive cells, cytokines, and small molecules within a liquid handler, dispensed into 96-volt plates and analyzed with a flow cytometer. My second aim was to predict factors which were important in engineering cell fate. To do this, we ran a predictor screen that cut down the number of experimental runs from 512 to simply 134. Then we were able to extract out which factors were actually important for adjusting HSC population. As you can see, it's TPO, GCSF, and UM171. In summary, engineering the HSC population allows for better patient outcomes after engraftment. Also, developing an automation pipeline increases speed, reproducibility, and reduces the error rate. Lastly, from this, the automation pipeline, the generated models are able to predict factor combinations for improving patient outcomes. Thank you.
Hey everyone, my name is Stephen Zhang, and today I'm going to tell you how to produce nozzles for single-cell printing. First of all, why do we need single-cell printing? Even cells originated from single cell can have different genetic composition. So it's going to be nice if we can separate each and individual one of the cells and do tests on each individual one of them, especially in fields like PCR and stem cell research. But to get consistent cell count, we have to manipulate the nozzle's contraction angle. The nozzle's contraction angle is the angle formed by the nozzle's inner wall, as you can see from the right picture. And if the contraction angle is not right, the cell, as you can see from the bottom picture, actually gets pushed further into the nozzle instead of being printed out. And my job over the summer is to make nozzles with different contraction angles so that we can experiment on them in future. The first technique I'm going to go over is called nozzle pulling. So how that works is that you thread a capillary tube through a heating element. The heating element heats up and melts the capillary tube in the middle. At the same time, two clamps on either side of the capillary pulls it towards the opposite sides. This separates the capillary tube in the middle, allowing a taper to form, perfect for a nozzle. And this technique is very good to produce nozzles with very small contraction angles. But if you want a nozzle with larger contraction angle, you need a technique called pressure polishing. So how that works is that you connect the blunt end of the nozzle to an air pressure source. The air pressure source blows air into the tip of the nozzle, and at the same time, the Bunsen burner heats up the tip of the nozzle, softens it, allowing air to expand it. The contraction angle increases as a result. All in all, now we can produce nozzles with a contraction angle from 4 degrees all the way up to 68 degrees. And here are some suggested parameters. Because I only have 2.5 minutes, I can't go into too many details, so feel free to check out my poster at D48. And thank you so much for listening, and I hope you guys have a very nice day. Take care.
Hey guys, I'm Tom and I'm going to talk a bit about cyclic peptides today. So bacterial biofilm is composed of proteins, polysaccharides, and extracellular DNA, and contributes to almost 2/3 of all clinical infections, and confers 10 times to 1,000 times fold increase in antibiotic resistance. So the host defense peptide IDR1018 has been shown to have broad spectrum anti-biofilm activity as well as immunomodulatory functions like suppressing inflammation, but lacks trypsin stability and tends to aggregate in vivo. The cyclic peptides I'm going to be talking about today have shown increased trypsin stability and also have low aggregation profile. So we're trying to generate these derivatives with improved biological activities as well as physical characteristics. So these 14-mer cyclic peptides are linked through cysteine-cysteine disulfide bonds, and single amino acid substitutions were made with hydrophobic residues, glycine, and positively charged lysine and arginine, with 96 peptides in total. So the free-swimming planktonic growth inhibition was seen to be most effective with phenylalanine, tryptophan, and valine substitutions against MRSA, but against some gram-negative strains tested like E. coli and Pseudomonas, they were less effective. For the biofilm growth inhibition, phenylalanine, tryptophan, lysine, and arginine substitutions were found to be most effective against MRSA at the concentrations tested, and they also had improved effectiveness compared to the parent compounds. Looking at the inflammatory response and immunomodulatory functions, the monocyte chemoattractant protein 1 production was varying across all the peptides. And IL-1 beta production after LPS treatment, there was little suppression with the peptide treatments. So in summary, screening 96 peptide derivatives, tryptophan and phenylalanine substitutions were found to be most effective for planktonic and biofilm growth inhibition, and suppression of inflammatory cytokines or CP1 were less effective. These peptides will be further studied for various aspects like antimicrobial, antibiofilm, synergistic effects, as well as cytotoxicity. Thanks everyone.
Hi, I hope most of you are still awake. My name is Valeria, and this summer I worked in Oryx's lab, where I synthesized new non-macrocirculators for nuclear medicine. In particular, we are working on radiopharmaceuticals, and these are compounds that use radioactive isotopes for therapy and diagnostic, depending on metabolism, radioactive decay. And on the right is a very famous example that shows the power of this technology in treating cancer. And in this case, I believe this is using the actinium isotope. So the general structure of the radiopharmaceutical contains 4 main components. And first is the radioactive ion, which is a source of radiation used for diagnostics and imaging. And then we have the chelating ligand, which our group studies, and it encompasses the radioactive ion, making sure that it does not escape the combustion. And that is connected to the targeting vector via linker. And the targeting vector is what makes this therapy so powerful, because it is targeted and it will deliver the compound to the desired site within the body. For example, cancer cells overexpressing a certain marker. So we are mainly working on the non-macrocyclic ligands that offer several advantages over the macrocyclic ligands that are currently used in most of the clinical trials and approved radiopharmaceuticals. And these advantages include faster coordination, meaning that you don't have to sacrifice the half-life of the isotope, and also milder conditions that are more compatible with the targeting vectors. And at the bottom, the two ligands were the ones that I have synthesized in the lab, and I have tested them with two different metal ions. So first is the noctable ligand, and here you can see that it forms a metal complex with a lead ion at pH 2 and 6, where you can see the splitting patterns that indicate the change of the chemical environment as opposed to the free ligand without the metal present. And we also predicted its structure using the DFT, or density functional theory, and it forms a really nice symmetric complex. And then the next ligand, the next ligand is the H2 noctiva, and I formed a complex with lead, and here again you can see the split pattern that indicates the complex formation at pH 5 and 7. And same is seen with the indium ion. So these are very promising results that tell us that we can take these compounds and try to radiolabel them and quantify the results to see if we can incorporate these chelators into the radiopharmaceuticals. Okay, and yes, see me at the poster 45 if you want to talk more about the radiopharmaceuticals.
And today I'm going to be sharing my project that I worked on in the Kazakh lab. My project focused on developing a new method to quantify iron levels in vitro iron overload models. So red blood cell diseases like sickle cell anemia affect over 300,000 babies born each year. And these patients require frequent blood transfusions, which results in harmful excess iron being introduced into the body. Chelators are used to bind these free irons, and this helps as treatment. Currently, the gold standard way to quantify total iron is by inductively coupled plasma mass spectrometry. However, there's no way to quantify free iron or the labile iron pool. Therefore, for the summer, my project was to develop a new method to quantify the labile iron pool by colorimetric method. Our recipe has 3 main ingredients. Because iron is found in 2 different forms in cells, represented by the pink box and the green star, ascorbic acid was used to reduce iron from one form to another. This is because ferrine, which can be detected by colorimetric technique, can only bind iron in one form, represented by the green star. Ammonium buffer was used to maintain pH for ferrine, so that it's able to bind iron. As you can see, the 3 lines at the top represent iron added to our 3 solvents: water, ammonia buffer, and lysis buffer. The 3 lines at the bottom have chelator added, and the lines in the middle have chelator with iron added. We were able to determine an ascorbic acid concentration that is required to reduce enough iron so that it's detected by ferrine. We tested this in our 3 solvents and found that the solvents do not affect ascorbic acid's ability to reduce iron, which is great because in any change in iron levels detected is actually a change in iron concentration. We then used to determine the ascorbic acid concentration and measured iron levels in iron overload cells with and without, treated with and without chelator. And as you can see by the yellow graph, there is no significant difference seen in the total iron quantification, and this makes sense because the cells were treated with the same amount of iron to begin with. However, when looking at the green graph, there is a significant difference seen in the labile iron quantification, and this also makes sense because when chelator is bound to the free iron, it's not able to be detected by ferrine. Therefore, we were able to conclude that our simple, low-cost method to quantify the labile iron pool does indeed work. If you have questions about my research, please feel free to drop by poster 46. Thank you.
Good afternoon. My name is Yalda, and I'm from the Stranatka lab. One of my projects this summer has been to structurally characterize the divisomal scaffolding protein DENT. As many of you know, cell division is one of the most fundamental processes required for life. And bacterial cell division is facilitated by the divisome, which is a ring-shaped organelle made up of over 30 proteins. However, many of these proteins have yet to be characterized, and DED is a scaffolding protein that has been shown to promote synthesis of septylpeptidoglycan by activating downstream proteins. DED is a bitopic intermembrane protein that has 5 highly variable helical regions, shown in blue, and a spore domain shown in green. I'm currently focusing on the soluble construct, which is underlined. I'm expressing DED in an auto-induced media, and I'm purifying using nickel chromatography and size exclusion chromatography, and I'm screening in over 1,000 unique crystal conditions. As you can see highlighted in red, we are able to obtain a high yield of relatively pure DED. However, lack of a single peak in the size exclusion chromatogram suggests that there may be disorder in the H1 to H5 helical regions that I previously mentioned. There are no crystals that have been obtained as of yet, possibly due to this disorder, and as a result, we're currently looking into crystallizing just the spore domain. If you're interested to know more, please come see me at poster number 47, and thank you for listening.
Hello everyone, my name is Yuyan, and I'm from the Brown Lab. My project is on the validation of a 20-gene signature that demarcates different types of small vessel vasculitis in children. So what is small vessel vasculitis? SVB describes a group of diseases characterized by inflammation of small blood vessels, and they can occur in both children and adults. Conditions caused by SVB can be life-threatening because they often target vessels in brain, kidneys, and lungs. Two major subtypes, referred to as GPA and NPA, are difficult to tell apart but will likely benefit from different treatments. The problem is that the current classification tools are based on symptoms and are so imperfect that GPA and NPA are treated the same. RNA sequencing on whole blood collected from SVB patients at time of diagnosis subdivides them into 2 groups, which can be seen on the right in the correlation heat map that denotes the GPA and NPA-like clusters. The disparity in gene expression between the 2 groups can be distilled into a 20-gene signature, and the aim of this project is to validate the signature for SVB subtype classification. Gene expressions were evaluated in 4 cohorts by performing reverse transcription qPCR on RNA samples. A healthy pediatric control cohort determines the inter-individual variation in gene expression and set the baseline expression level. A large vessel vasculitis cohort to determine if the signature is specific for SVB. A discovery cohort consisting of SVB samples that were used to generate the signature to confirm the RNA sequence results, and finally a replication cohort of SVB samples to determine if the results can be generalized to other SVB patients. In both replication and discovery cohorts, qPCR results showed that the expression level of 20 genes are different in GPA and NPA and between these groups and controls. Of these 20 genes, 10 genes may be specific for SVB because we did not observe any change in expression level in large vessel vasculitis. In summary, my results show that there was consistency between RNA sequence and qPCR results and between SVB samples in discovery and replication cohorts. A subset of the genes that may be specific for SVB was determined, and future work involved assessing if the signature is retained over time. Thank you, and for more information, come and see me on poster 49.
So that brings us to the end of the student presentation, so I think we should thank everybody for presenting. Thank you very much. So I suggest everybody to sort of get up, give a little stretch before we move on to the next. You can do your voting. Oops. So you can do your voting as well. And I'm going to call on Stephanie. Come on down. You can talk, you can mumble. Before we move on to the next part. This is the 7th inning stretch. Well, just make sure you come back down. Okay. Okay, don't go away, don't go away. Okay, everybody, let's go. We're going to start.
All right, I think we're gonna get started now. All right, I think we're gonna get started now. Hi, everybody. I'm excited to introduce you to this year's Research Day keynote speaker, who's coming all the way from the East Coast of Canada in Dartmouth, Nova Scotia. He currently works for a coalition of environmental nonprofit organizations in Atlantic Canada with the shared goal to protect ocean biodiversity. Previously, he was the managing director of biodiversity at the Royal Ontario Museum, as well as the senior curator of environment at the Toronto Zoo. And as you can tell from his experience, he enjoys working at the intersection of science, policy, and public engagement for the conservation of nature. And I'm sure his talk today will also reflect this. Please help me welcome Dave Ireland.
Awesome.
We're good to go, I think.
Okay, I don't think I need the microphone. You guys all right back there? Okay, so before I set my timer, because you guys are so on the ball with time, I don't want this to be included in my 25 minutes, but I did a bit of judging myself, and you guys were amazing. Like, really amazing. But I did choose my top 3 and a runner-up, so I think I just want to do that and see if I can influence the broader judging schematic a little bit. So these top 3, I think all did sort of 3 things really, really well. They had energy, they knew their content really, really well, and they brought me, who is not a blood scientist or a genetic engineer, into the conversation a little bit more. I felt like I understood what they were saying. So sort of first one, Jessica, I don't know where she is, Jessica Ha. That was amazing introduction using your grandmother. I thought that was brilliant. Paige Yo, the storytelling and the real world application was just fantastic. And then sort of one of my favorites for sure was Sia John Abu. Wow, what great storytelling. You were amazing. I was like kind of sitting back for a while, and then she got up there and I was like on the edge of my seat. Amazing. And the runner-up, of course, was Meredith Clark, because she got just scammed there with time, and you still pulled it off, and you were really, really well spoken. So you guys are in for something totally different than what you've just heard over the last couple hours, and I hope you make connections. I certainly feel like there are connections to be made in terms of storytelling, science, the scientific process, how people use the scientific process, and how it's applied to real world situations. So that's what I'm going to do. I am going to start this now, so that was just 2 minutes. Oh, sorry. Perfect. So I may or may not skip out on a couple of these stories, but certainly that's the 4 stories. They're kind of interconnected, but I'm going to flow between them pretty seamlessly, I hope. So I grew up in Huntsville, Ontario. Anybody know it? Yeah. Anyway, it's the gateway. There was one guy up there. It's the gateway to Algonquin Park, and certainly my first job was leading canoe trips in Algonquin Park and being around nature, and the salamander was what sort of captured my scientific imagination. So I did my first degree 3,000 kilometers away from Huntsville, largely to get away from my parents. But I did a degree in biology, started thinking about science and biology and processes, and did a minor in chemistry. And then I took 5 years and I sort of traveled the world using tree planting money. And I came back to sort of Canada with the goal to do graduate work with these salamanders, amphibians, and reptiles. I landed in another university and I started studying the sort of the connectivity between populations of salamanders. I was using genetic techniques, so there was a little blood involved. I was taking blood out of these salamanders and frogs. And then I started asking bigger questions, and I went down this real rabbit hole, this academic rabbit hole, which I kind of feel like you guys are all in right now. So all the power to you, but when I was publishing papers with titles like Phenotypic Plasticity of Ranoclinitan Embryos Under Varying Predation Risks, I was surprised my readership was so low. And so I started to ask myself bigger questions around how is my research, my PhD work, how is it actually gonna change the world? And obviously I don't need to tell you guys this, but we're in the midst of a biodiversity crisis. And I'm not going to get into that. This will be a very hopeful presentation, I hope. But I took a complete 180 degree. I sort of stopped academia, I stopped teaching, and I moved south of Highway 7, which I said I'd never do. And I went to the Toronto Zoo, and I started a new position for the zoo itself, and basically the position was to make the bridge, connect the bridge between the scientists that were in the back of the house at the zoo and the public. Because generally 1.5 million people go to this particular zoo every year, and they never see the 10 scientists in the back or the research associates or the postdocs or the vets. So my job was to make them somewhat famous. I have a ton of stories about that, but I'm not going to talk too much about it. I just want to tell one quick one, because it has to do with salamanders. So again, I was doing very academic work with salamanders, but when I went to the zoo, I started thinking about salamanders in an applied sense, and I found, through some help with some students, a population of very endangered salamanders in Burlington, which called the Jefferson salamander. And they were literally, their predominant threat to their existence was a single little arterial road that commuters took to get from their bedroom communities into Toronto every day. And the population of people on that road wasn't large. So we approached the city of Burlington, this is 10 years ago, 2009. We approached the city of Burlington and said, listen, if you allow, because these are explosive breeders, they breed for 3 weeks. Then they're done. Then they're mole salamanders, so they go underneath the ground for 11 and a half months. So for 3 weeks, that's all we need to do is close this road down for 3 weeks. And the city laughed at us and said, are you kidding me? People? Salamanders? Nah, that's not going to work. We advocated and advocated and advocated, and they allowed us to do it, but we had to staff the road every day. We had to explain to the commuters who were PO'd why they couldn't do this road. The first year was hard. The second year was a bit harder, not as hard, got easier and easier. And then we stepped back, and the city of Burlington now owns this, and not a single salamander has died on that road in 10 years. So that's a great story. I did promise my wife that I would not be in Toronto for more than 5 years, but another job came up at the ROM. So I took this job, and we spent another 8 years in Toronto. And the ROM is a bit different than the Toronto Zoo. Toronto Zoo, there might be 10 scientists, quote unquote. At the ROM, there's 65 curators. Kim Tate holds the biggest collection of Mars on Earth. Burton Lim is probably the most authoritative bat biologist in North America. This guy's publishing a paper in Science and Nature every 3 to 6 weeks, describing a new species. He's the authority on the Burgess Shale. So heavy hitters, but the job at the zoo, or the job at the ROM was the same as the one at the zoo. What they wanted me to do was make these people, and with all due respect, people like you, really interesting to the public. And I do need a job right now, so you guys are not coming west. So at this point in my career, I've been doing a lot of this stuff on my own, raising grants, thinking about how to do it, and and learning social media and learning communication tactics on my own. But then I got to the ROM, and there was this huge amount of people. So I started to think about, well, I can't do this on my own anymore. So for example, this is just one quick story that sort of segues into the true message of this bit, which is environmental visual communication. So this is Doug Curry. And I chose him intentionally because Doug Curry studies black flies. What do black flies like to eat? Everybody together? Blood. So Doug wrote this huge book on black flies. He studies black flies. Nobody likes black flies, yet he was a prolific world leader on this sort of topic. And the question was, how do we make him more interesting? So it was actually, his graduate student, his postdoc, Santiago, who came out with me for a beer one night. We were talking, how are we going to do this? And Santiago came up with the idea. He was like, all right, on Halloween night, let's keep the museum open like another hour or two. And we had some funding, so we paid for the security and everything else. And we'll just put Doug in the middle of the museum with a big jar of blood. And it was literally about 9 liters of blood and a microscope with some black flies on it. And that was it. No sign, no nothing else, just blood and the black flies. And then we allowed people to drink, which was very helpful. And so that spawned Doug into being able to talk about his work in the 2 minutes he was able to keep their attention. And the blood got them there, the microscope allowed them to tell a quick story, and then they were off. In so doing, the reason this slide's up is Doug then embraced Twitter. He was the first curator at the ROM to really embrace Twitter. This is before the hashtag SciComm really came out. And now Doug, this is a very old slide, so that's from 2012. He's now pretty prolific on Twitter. At the end of the day, what this was telling me, it wasn't me that came up with that idea. It was Santiago, who was 15 years younger than me. And it was this younger group of people, 25 to 35-year-olds, who were coming up with this innovative way to communicate science. So what I did on their behalf was I came up with an idea where we could create more capacity. So we built a graduate level program called the Environmental Visual Communication Program. It's one of a kind in the world. It's a boutique program where 20 students are permitted to come to the ROM every year for a 9-month intensive study of photography, videography, social media campaigning, design principles. I taught the oral and writing and communication components of the course. They couldn't work. It was 50 hours a week, and they would learn intensively how to communicate science. And so to date, we've graduated 120 students, and 75 to 80% of them have jobs in the sector. And these people are now totally geared up and capable of working for groups like yours or for other people, and indeed that's what they're doing. So what I'm going to do now is I'm just going to tell you a few quick stories about how these students have transformed the way at least the ROM is telling their story. So this one again is an evening program at the ROM. We invite, it's called Friday Night Live. If ever you're in Toronto in the spring, I would seriously consider attending an FNL. It's a huge party. About 3,000-4,000 people come into the museum at night. We allow drinking again. God, this alcohol thing. But basically one of those nights we had a team. I was project managing a team of 9 scientists in Borneo. They were studying the taxonomy and biodiversity in Mulu Mulu, which is an area of central Borneo. And they're 12 hours different than us. So what we did, again, student-driven, is we put a student in the field with biologists for the 3 months they were there, and they choreographed a live stream back to the museum during this FNL. So it's 9 o'clock our time, everybody's loose and having fun and dressed up, and the curators are all in the field at 9 in the morning drinking coffee, and it allowed them to speak in real time with these people. Most of them were happy about it. Chris, not so much. We've sent students to Sri Lanka, and here's just a couple pictures from that. It's actually one picture. And then finally, this is sort of my culminating story with the EVC program. It was 2014. For reasons I'll describe in a second, there was a mass mortality event of blue whales in the Gulf of St. Lawrence. 1% of the remaining population on Earth died all at once in the Gulf of St. Lawrence, and nobody knew why. We got the call from the Department of Fisheries and Oceans the next day saying, do you guys want these whales, or at least 2 of them? Because they were fighting the province, Newfoundland and Labrador, and the DFO were fighting, right? At low tide, the whales were the responsibility of the DFO. At high tide, it was the response. So every 6 hours, this was changing. And so they were fighting. They asked us if we could help. We raised a lot of money very, very quickly and sent a full team to Newfoundland to collect these whales. And so this is just a series of photographs that will describe to you how that was done. Very quickly, it was actually quite a popular thing. I don't know if any of you guys remember it, but when these pictures came out by our student Jackie, they went global. BBC, every news outlet in the world was showing this particular photo, and the question was being asked: Is this blue whale going to explode and kill people or hurt people? It was preposterous. Saturday Night Live called us and they did a bit on it. The whale deflated, right? It doesn't blow up. You know that. And so then we towed it to shore. There's Burton again, one of North America's foremost bat biologists. He doesn't study anything bigger than a fruit bat. So he was none too pleased when the boss said, you're going to Newfoundland to cut up a whale, but he did it. The students documented everything in Newfoundland, and then we brought the whales back and put them in manure for a year to decompose them. The students documented that whole process. They documented the process of cleaning it. They documented the process of 400 hours of cleaning the baleen from 2 animals. They cleaned the baleen from 2 animals. These students who were paying to be environmental visual communicators were cleaning baleen. That's another story, which they actually participated in one really remarkable project of the blue whale story. Does anyone know what this is? Tongue, I heard. It's not the tongue. I heard it. It's the heart. So we basically, I don't know if you guys saw it, but 5 years ago, Sir David Attenborough got on one of his shows, Blue Ocean or something, and he said that the blue whale heart, the arteries in the blue whale heart are big enough for an adult human to swim through. And David doesn't say anything unless he is pretty sure of it, but no one had ever collected and preserved a blue whale heart. So he was saying that based on models of finbacks and humpbacks and extrapolation from size differences between those animals. And he was wrong. We were the first to actually preserve, and this is the real thing. It's a real blue whale heart. It's now touring through China right now. But I could get my head through it, maybe one of my shoulders, but not my whole body through the biggest vessel. So we tweeted David Attenborough, said, hey, what's going on? And he actually tweeted us back and said, oh, thank you. And so we played a big role in sort of that sort of mass communication as well. Long story short, we made a big exhibit on this. We produced this exhibit in 2017, and then we sold, well, we didn't sell it. We're loaning it out for like $400,000 every 3 months to different places all over the world, and it's currently touring through China. So I'm not segueing too far, but part of my role with the Blue Whale Project, and I am going to try and answer this question, can whale poo save the planet, was, I'm a conservation biologist, so my role was really to provide content for the exhibition and the project overall on the status and answer the question, why did these 9 blue whales die in the first place? Our public coming to this exhibition, and a lot of people came to it, and a lot of people are seeing it now in other places, are asking the question, well, why did they die? And it is an endangered animal. It's not endangered because we were harvesting them for oil or for corsetry or for any reason at all. In fact, this is the only doom and gloom part of my whole talk. We didn't actually learn how to kill a blue whale effectively until after the world signed the moratorium on killing of large whales. So in 1965, because of protests and advocates, we couldn't kill whales anymore. It was redundant. We didn't need the oil. Petroleum had taken over. We didn't need the baleen. Plastics had taken. There was no use for large whales other than maybe dog food, and so there was protests, and we stopped killing them. But we hadn't figured out how to kill a blue whale until about 1967. They're big, they sink when you kill them, and they're very, very fast, fastest whale out there. But humans are, as humans are, we innovated in between 1966 and 1969. We figured out how to kill these whales. And then during the moratorium, during the period where the world had agreed not to kill any more whales, we killed 99% of every blue whale on the planet in like 8 years. It was the biggest slaughter of megafauna in our entire history. And we did it after we said we wouldn't. So I will bring it back up. So this is what we killed them for. This is the brunt of the joke. So most blue whale meat, most blue whale parts were used to make soap between 1970 and 1985. Horrible. Anyway, back up again. My job was to figure out why in fact these 9 blue whales died. So we didn't have a good cetacean biologist on staff at the ROM, so they sent me and a team of students, again the EDC students, to Quebec, to the North Shore of the Gulf of St. Lawrence to work with scientists in the field and ask the question of these scientists, why did those 9 blue whales die? And I got to go on a zodiac. I'm not a whale biologist either. In fact, I study salamanders. But I got to go 25 kilometers offshore out in the middle of the Gulf of St. Lawrence. I couldn't even see land. At one point, we counted 50 humpbacks, well over 200 minke whales, right whales, finback whales, and blue whales all around us, almost at the same time, within the same field of view. It is an amazing place for whales to eat, the Gulf of St. Lawrence. I got very close to a 70-ton finback whale. And these guys are serious biologists. The Mingan Island Cetacean Study, they've been studying blue whales for 25, 30 years. Richard Sears is their principal investigator. He's world-renowned for his understanding of blue whales. So we knew, as the ROM, we needed the best source of intel, and we found it in these guys. They take biopsies, they study the genetics, they study sort of the scarring, they study the movement patterns, and they also tried to figure out the answer to that tricky question. Why did these blue whales die in 2014? Anyway, here's the answer. In 2014, first of all, how shall I start this? The Gulf of St. Lawrence, due to climate change, is the fastest warming body of water on Earth. It has been warming very fast for the last 50 years, and we have great data to show that. So park that for a second. Gulf of St. Lawrence is warming. 2014, and as all we all know, climate and weather are not the same thing, right? 2014 was an extraordinarily cold winter. 96.5% of the Great Lakes froze over. All of Lake Ontario, except for this tiny little pocket around some effluent pipes, frozen. 2014 was a cold, cold year. So Gulf of St. Lawrence is warming. 2014 is an anomaly. Now the whales. The whales for millennia have been coming up the eastern shoreboard, and they wait right there at the mouth of the Gulf of St. Lawrence to get in, to eat. And they come in like February and March, and they wait patiently until the ice breaks up, and then they go in there and they eat for 6 months straight, depending on what species it is. And then they leave and they do their thing. But as this Gulf of St. Lawrence has been warming, we have great data to show, especially finbacks and blue whales, are going into the Gulf of St. Lawrence earlier and earlier and earlier each spring. Why not? If the food's there and there's no ice, I'm going in. 2014, it was a massacre. Not just blue whales, but there was a crack in the ice. Whales were waiting all out here. They followed the crack in the ice to get into the Gulf so that they could eat. Crack froze up behind them, and a mass mortality event of cetaceans happened in the Gulf of St. Lawrence. So those whales died because of climate change, without a doubt. And we said that, and that was the first time the Royal Ontario Museum had ever used the term climate change in an exhibition? Sorry, whale poo. And you know what? I'm gonna gauge this audience in like 5 minutes to see where you're at, because I might not get to the final story. But so another part of this was, you know, the people who would come to this sort of exhibition would also ask the question, why should I care? What's the big issue if there's 400,000 blue whales or 1,000 blue whales? What's the big deal? You know, how does it impact my life? So we went down this rabbit hole of asking the question, well, what is the benefit to having large whales in our oceans? And they're plentiful. In many systems, they're keystone species. In many systems, they're regulating prey. But one of the predominant and unnoticed and really cool stories about the big whales is their poo. So think about, and again, you need to just sort of get your head around a few things, which you all are very capable of. Phytoplankton in our oceans contributes as much or more oxygen and carbon sequestration as every terrestrial plant on Earth. So phytoplankton are huge, dominant, and very important to regulating our climate. Phytoplankton are pelagic for the most part, right? So they're out there in the middle of the ocean and they have a few limiting resources. The sun, a few different types of nutrients that are fairly ubiquitous, and then some nutrients that are very concentrated, like iron and a few other things. Inside whale poo, and especially blue whale poo, just to give you a sense of scale, that's about 60 meters long and about 10 meters wide, and that's one movement. And that whale is pooing probably 70 to 90 times a day when it's feeding. That's one whale. So we did some extrapolation up from there, and then we asked the question, if there were 400,000 whales, blue whales, versus 1,000, what would our climate change adaptation potential look like? Then we asked the question, well, what about humpbacks, bowheads, which we slaughtered, right whales, which we slaughtered, finbacks, which are way down, all of the whales are way down. What if we took all of those numbers, pre-industrial whaling and to post-industrial whaling, and we asked the question, okay, and again, I think you found the loop, right? Inside that plume is the nutrients needed for huge phytoplankton blooms to grow. Those phytoplankton blooms do grow concentrated around whale poo plumes, and therefore we're increasing the opportunity for carbon dioxide sequestering and oxygen production. Anyway, long story short, we did this whole section in the exhibition on the whale pump, the story I've just told you, and we showed that very nicely in really concrete terms and language that is understood by the public, that whales contribute significantly to managing our climate. And if we could conserve them or in any way increase the abundance of whales, we'd actually be increasing our mitigation potential for adapting to climate change. So we put krill on display. That's real whale poo that I collected in the Gulf of St. Lawrence. And that's me in a krill suit. So the whale we put on display was all real bone, but we modeled the mouth of the animal and put that in a corner of the hall and opened it up, and you could wear the krill suit and get into the mouth of the whale and take your pictures. So. All right, so listen, I could do this one without turning a slide. Because I'm at like 26 minutes, so, and I don't want to, I don't want to do it, but you guys, none of you did, which is amazing. So my next big move right now, I'm living in Halifax, Nova Scotia, Dartmouth, Nova Scotia, and I moved away from hard science. I moved away from working directly with academics. And now what I'm most interested in is participatory research and citizen science. The premise of this, and again, it's not too doom and gloomy, but you guys should know this, or you should embrace this. The world is an incredibly complicated place right now, politically and environmentally, ecologically, and it's going down quick. And one tactic that a bunch of us really feel strongly about, and there's good science and evidence to suggest this works, that is, if we don't connect with the values and the interests of everyday people who don't understand a word of what was said earlier, the way they will vote will impact the work that you do. funding to science, policies for environmental protection, all of these things. This is why the populist movement is working, because they are catering to the values of these people. One way for us to do this is through something that we call citizen science. And, you know, at the end of the day, it's pretty simple. You basically create protocols and mechanisms by which the general public can participate in your work. And to the person who accepted the award earlier, I couldn't agree more. Mentorship and tutorial and inclusion of people who don't understand what you're doing is so much harder than just doing it yourself. No doubt. But if we collectively don't, your work might not be that important anyway. We need to get beyond that, and we need to open our camp. So what I've done in a long, winded story short, is I've created a program, a global program now, which started in Toronto, called the BioBlitz. E.O. Wilson coined the term in the year 2000. It didn't really take off until 2009, 2010. And in 2011, we created a program in Toronto called the Ontario BioBlitz program. And basically, long story short, we do intensive surveys of biodiversity in a given area over some set period of time, usually 24 hours. And we include the public in that work, and the data is used and brought up to GBIF and Canadensis in large, very reputable data sets so that scientists can use it down the road. If I were going to sort of transition from this slide, this is not what a BioBlitz looks like. This is what scientists think it looks like. That's part of it, but it's a very sort of structured routine. Two of these guys have published books, discovered species, all based on citizen sciences gathering the data. There's a spider within 1.3 meters of everyone in this room right now. Spiders are everywhere. But to collect them, identify them, and quantitatively assess their abundance is tricky. So these guys embrace citizen scientists to do that. The federal government gave us $1.5 million to take the Ontario BioBlitz program to Canada, and we launched a program across coast to coast to coast in 2017. We developed with our partners in the States an application just for Canadians. It's called iNaturalist. It's based on the iNaturalist app in the States, but it's bilingual. We're introducing the Mi'kmaq language for Atlantic Canada. You basically take a picture of something, and within a few minutes, if it's a common vertebrate, you'll get a species ID back. A little less, some more lesser known things, invertebrates, etc. Maybe it takes a day, but it's basically an application that helps you identify natural history. And again, all that data goes straight up to some very serious databases. And here's just one that will resonate with you guys. So this graduate student from Kentucky, he's a PhD student studying in Kentucky. He really likes this dragonfly. And he lives basically in the Midwest, and he's noticed that in one part of this dragonfly's range, the wing coloration is different than in another part of its range, a more northern latitudinally part of its range. And he had a hypothesis that the reason the coloration of the wings were different was because climate was changing, and he would like to show that sort of in an empirical study. But he needed data. And his supervisor was like, there's no data on these guys. And they looked through all their serious databases. And then an undergraduate from the lab said, just for fun, why don't you check out iNaturalist? They might have a few data points on this particular dragonfly that you could use to answer that big question. He went into iNaturalist, 13,500 very good, verifiable research grade observations of that species, all open source. He just dragged it out, put it into his model, and published in PLOS ONE that said, yeah, this dragonfly species is changing its range because of climate change. All based on citizen science. Anyway, final quick story is I needed to make an impact in Nova Scotia quickly, because again, I need a job. I actually have a job. I'm working for, like you said, Steph, I have a job. I'm just looking to, continue. Anyway, this will be my final story. So Leela from the Natural History Museum in Los Angeles, and her friend Allison from the California Academy of Sciences in San Francisco, LA. They hate each other, they love each other, they love to fight, they love to compete, And so Leela and Allison said, this is 4 years ago, said, why don't we do a fun little city-to-city challenge, LA versus San Francisco, one weekend, who can gather the most natural history observations, research-grade observations, and we'll see if we can raise the awareness level for iNaturals. They did it 4 years ago. The media loved it. It went sort of viral in California and the West Coast. And so the City Nature Challenge was born. Fast forward 5 years, a Canadian city has never participated until this past winter. And Halifax, yay! We did it. This line represents about, 30 other cities. So we didn't rank in the top 10 or anything, but we did pretty well. We actually won for lichen species. Did any of you guys know that Nova Scotia is the lichen hotspot of the world? Like, we have more lichen species in Nova Scotia than anywhere else. Latitudinally challenged cities, they felt bad for a few of us, so they made this like little subcategory, and we won it. There was only like 4 cities in the category, 3 of which were Canadian. And then we did really well because, believe it or not, Atlantic Canada is not so populated. My favorite thing that evolved, this is my last slide, my favorite sort of thing that evolved from the City Nature Challenge, the BioBlitz movement. I've been working on this for almost 10 years now, and it's growing, and it has some serious legs and serious applications for other people's work. You can take what we did in a BioBlitz and apply it to blood research or any kind of scientific endeavor. It will require more work. But here's a really good feel-good story to leave you with. This Girl Guide troupe from sort of northern, or they never call it north, what do they call it, eastern shore of Nova Scotia, approached us and said, we love iNaturalists, we want to do the City Nature Challenge, but the girls, the contributing participants of this little troupe, they want something in return. Would you guys mind making a badge? So I called on some old friends at the ROM. I said, hey, would you guys fund a first order of badges? The ROM said yes. Canadian Wildlife Federation established the curriculum for it. And just before these girls went out for City Nature Challenge, we presented them with these badges. And it was amazing. These kids, they were over the world happy having got these badges. And now Nova Scotia, the first little jurisdiction in Canada in the world to do this, it's just gone viral. Calgary wants to do it. California wants to do it. All over North America is interested in this idea of increasing Girl Guide contributions to natural history investigations. So, really nice story. Thank you.
So we'll take a couple of questions.
Oh, nice.
From the audience.
I'll leave that one. No questions. No problem.
But we'll have lots of time.
Yeah.
And we're drinking.
Thank you guys, and thank you for inviting me here. Oh.
Heck away.
Oh, fun.
We have some thank you gifts.
Oh, fun.
We'll do a tote bag with some goodies inside there, and then a flag.
Chopsticks.
There we go. Thank you.
Awesome. Nice.
Okay, so we're wrapping up. Okay, we're gonna go drink and eat. Okay, so there's food out there. There's drink. There's posters. So you're the poster judges and poster people, students. There'll be judging going from essentially now until about close to 5:30. Make sure all of you do your voting, and just you can just leave them here or pass them to the end, and we'll collect those. Everybody should make sure you do your voting. The summer students, there are T-shirts. Okay, there are T-shirts to be picked up. along with other goodies, I'm not sure. Anyway, we'll buy from Hannah and from Stephanie at the registration desk as you walk out. There'll be pictures as a group of the students and the graduate, the GAP students, the new GAP students. Please make sure that you're around so we can take pictures of you as a group. What else? Other things, requests from the administration in particular. Clean as you go, please, because there's a big cleanup effort at the end of the day, because you know it has to be spotless for tomorrow morning. So just as you're going, just think, maybe I'll just throw that beer can where it's supposed to be, okay? And then you're welcome to give feedback on this event and, of course, any other event. I think that's it. That's it. Thank you all very much.